Here you can see the pair complex stage, and it's a critical intermediate stage in this process of recombination. What happened here is very simple. You have the DNA segment here, and what you find here is this. You have the RAC enzyme here with two spacers. Yeah, one spacer for the G segment and our longer spacer for the B segment. Why? Because remember we want the proper alignment, yeah, and this is ensured by having spaces of different length. And now what is going to happen, if you see here, uh, is that they are going to cut this signal. Okay, so I just show you that the recombination machinery brings together two recombination signals, the RSS, and here these recombination signals have to be different, one from the B section and the other one from the G segment. And why this is important? Because it ensures precise alignment of the segment that facilitate the proper recombination. Here, what you can see is the following two steps, and here you can see a summary in using this diagram of what is happening. So in the first stage, you see that the enzyme, all these drag and different enzymes, what they are doing is recognizing the specific DNA sequence. Yeah, the recombination signal sequence, the RSS, that are flanking each one of the gene segment, B, D, and G, and producing this cut that you can see here. Yeah, so they recognize a specific place. And the second thing that is happening here is that you see that the joining of the segments. Yeah, so here you see that the DNA again is continuous. Yeah, so they are joining together the variable and the diversity if it's applicable, and they join the segment together. And how they do it? By using some enzymes like a ligase 4. Okay, so let's recap. So I show you that here in this stage there is something called a DNA cleavage, and here what you see is the RAC complex that recognizes the specific DNA sequence. So you find this RSS sequence that is flanking the B, the D, and the G segments, and that makes a cut in the DNA. The second thing that is happening here is the joining of the segments. So here you see what you found is that there is a DNA repair machinery that is joining the different gene segments and forming a continuous gene sequence. And I tell you that this can produce enzymes like the DNA ligase 4, as well as this non-homologous in joining, that is the NHEJ pathway.
So here in this diagram, I just want to summarize all the process that we have been talking. So here you have it for the light chain, yeah, and here you have it for the heavy chain. And you can see the difference that here there is diversity gene arrangement, and here there is not. Okay, so here you're missing that component. So this is the difference between the light and the heavy chain. There are more difference, but right now this is what we're going to focus. And here they show you what is happening at the DNA level. This is what happened at the RNA level, and this is what you're going to get. So look, here you're going to get these variable regions, yeah, in the light change, and the other one in the heavy change. And you can see that in the heavy change you're going to, in addition to red, yellow, you're going to get also those green diversity segments. And let's talk about what is happening here at the DNA segment. So there is somatic recombination. So you see what is happening here. That is between the D and J, and here is V and J or D and J. Okay, so and remember, as I said before, there is not one red segment, one green segment, one yellow segment. There are multiple, and the other one, the extra ones, are just recombined out, so they disappear, and they are removed from those new cells that are going to have that are going to produce this or antibody or receptors. Then after you get this new DNA sequence, you get to transcription, and here you have a primary transcript. There's splicing, and you produce this messenger RNA, and then you go to translation, and you end up forming the antibody that or the receptor that is going to form. Okay, so with this, I finished this section. I'm going to show you a video that summarizes each one of these steps. All this machine responsible of this BDJ recombination, that hopefully it can help you to remember each one of the steps. And if there is any question, you can rewatch this video. Hopefully everything was clear, and have a good day, and bye.
Immunoglobulin genes are composed of separated segments of DNA that become joined together by a process called somatic recombination to make a functional gene. In heavy chain genes, there are 3 gene segments: the variable, or V segment, a diversity, or D gene segment, and the joining, or J segment. Light chain genes, such as those shown here, have only 2 gene segments, the V and the J segments. Gene segments that can be recombined have specific sequence motifs adjacent to them, called recombination signal sequence, or RSS motifs. A protein complex containing the products of the recombination activator genes, RAG1 and RAG2, binds specifically to the RSS motifs, in this example flanking a V gene segment and a J gene segment. The individual gene segments, to whose flanking RSS motifs the RAG protein complexes bind, are selected at random from a number of copies present at each gene locus. The RAG protein complexes bring together the gene segments to be recombined and cleave the DNA exactly at the junction of the gene segment and its adjoining RSS motif. The cleavage creates a hairpin of DNA at the end of the gene segments, and double-stranded breaks at the ends of the RSS motifs. Additional proteins, DNA-dependent protein kinase, Ku, Artemis, and a dimer of DNA ligase and XRCC4 are incorporated into a large complex with the RAG proteins. These RSS ends are joined, forming what is called the signal joint, to create a closed circle of DNA that plays no further role in the recombination process. The DNA hairpins at the ends of the gene segments are then cleaved. An additional enzyme, terminal deoxynucleotidal transferase, or TDT, is recruited and adds additional nucleotides to the ends of the DNA strands. The other enzymes in the complex ligate together the 2 ends of the gene segments, completing the recombination process.